What's up, baby bio penguins? So it is Friday, FRQ Friday. Yeah, you know, I do this every week and we just sit here and go, why is she doing this? But so today we're going to do 3 different questions. They're all going to be short FRQs, and with them, I'm going to try to show you different ways that you can get points, even if you have no clue what's going on, right? Because sometimes we get to these questions and we don't know what's going on. So the goal today, I'm going to show you different ways that you could just use diagrams to kind of help you to get a lot of the answers. Okay, so hi everyone, welcome. We're going to go ahead and get started. So the 3 questions we're going to do today, we're going to do 2017 number 6. We'll do 2016 number 4 and then 2013 number 5. Okay. So our very first question, we have, what the heck is this? Like that's a crazy diagram. There's a comet assay. It's a technique used to determine the amount of double-stranded breaks in DNA. What? Wait, there's a comet assay? I never learned about this in my class, is what you're probably thinking to yourself. And that's what probably happened on the AP exam, because I never talked to my students about comet assays. And so a lot of times they're going to put things on the exam that you may never have seen in your class. Okay. So the point is that as soon as we see something we don't know, take a deep breath. I know this. I'm a penguin. I know this. I'm dressed for success. Okay. And then go back to your question and keep reading and see if you can come up with any information that might be able to answer that question, right? So it tells us that there's some damage, right? The nucleus of an individual cell is placed on a microscope slide coated with the agar shell. I've heard of that word before. I use that with gel electrophoresis. Sweet. Okay. An electric current is applied to the gel that causes the DNA to move, called electrophoresis. Oh, yes. This is just like the thing I did in class. And the DNA is stained with a fluorescent dye. When viewed using a microscope, the undamaged DNA from the nucleus appears as a round shape, the head. So I see right here, there's a circle. That just shows me undamaged DNA. And the fragments of the damaged DNA extend from the head to the tail. So I see that we move it and there's like this part right here, and that's my damaged DNA. The length of the tail corresponds to the amount of the damage. So the longer my tail is, the more damage must have occurred within the DNA. So if I read through this, it says, to explain the movement of DNA fragments in the comet assay, identify one property of DNA and provide reasoning to support how the property contributes to the movement during the comet assay. Okay. So number 1, when you see this, you can go ahead and give yourself some little tips in your head. Say, okay, what do I already know about DNA? Well, I know that DNA is nucleic acid. Okay. I know the DNA is made up of sugar, phosphate, and a nitrogenous base. Okay. What else do I know about DNA? Okay. I know it's double-stranded. Okay. What else do I know about DNA? And you can give yourself like a list on the side to try to come up with different properties that you know about DNA. Okay. And then from that, you're going to then give reasoning to say why it moved. Well, I don't know why it moved. Like that doesn't make any sense. Like the fact that it's made of these things, what does that tell me? That tells me nothing, right? So let's look at our picture. So if we look at our picture, we see right here, okay, that before we did the assay, there was just this circle, right? And then after we did the assay, we see that there's this circle, and then we see the tail, right? So by turning on the electrophoresis, by turning on this negative and positive charges, I can see that then the DNA is moving, right? So if you notice, the DNA is moving toward this positive end. Well, we all know that opposites attract, right? If you have something that's negatively charged, it's going to be attracted to a positively charged side, right? So right there, property, DNA must be negatively charged. Justification, because the DNA moved toward the positively charged end. Okay, and that was actually one of the points you could have gotten. You could have gotten a point for identifying that DNA was actually just saying that it had a charge. And negative was the optional thing. You see how there's those parentheses there? So if you ever see a parentheses on a scoring guideline, that's like an optional thing you could have said. So negatively charged, and then the DNA is moving toward the positive or an oppositely charged pole. Okay, so right there, all we did was look at a picture, and you got 2 points by looking at the picture and being able to make some connections and draw some information out. Okay. The second part, or the second way you could have gotten a point here, isn't as logical. So you would have said that they could have been different sizes. So the reason why we see a tail instead of a whole second head is because of the fact that they must be different sizes. And so when you run the gel, like what happens in the gel electrophoresis is there's these little pores in the gel. And the smaller fragments can move a farther distance. So the TikTok user, his name's actually a biology teacher, if you want to follow him. But he came up with a great example the other day in which he said, it's like you, your dog, are out for a walk, right? And the dog sees a bunny rabbit and you're in the middle of the forest, right? So that bunny rabbit can jump in and out of the forest, kind of burrows a lot faster than your dog, right? And your dog can run really fast, right? But then you are kind of held back by the bushes, right? So the longer fragments are not going to be able to get as far in the gel because they can't get in and out of those pieces, right? And the bunny rabbit, or the smallest fragments, can get the farthest because they're able to get in and out of those pores a lot faster. So I see there's a question. So with it's really hard to know how in depth to go with questions. So a lot of times when you're doing free responses, I just say to answer the question. There's a big thing that a lot of AP teachers say. It's called ATP, answer the prompt. So if your prompt is asking you to describe and do this whole detail, then yeah, give it that detail. If it just says identify, you can do a one sentence and you're probably there. And I've also, like, identification points, a lot of times you don't actually have to write a complete sentence, but I always tell students to write a complete sentence. It takes an extra 10 seconds to write "it is" in front of whatever you're identifying. So I would definitely write as a complete sentence, DNA is negatively charged, as seen by the fact that DNA can move farther. Hey, Elizabeth, DNA can move towards that positively charged end. So that was part A. Part B, it tells us that there's a different experiment in which the cells are treated with a chemical mutagen that causes only nucleotide substitutions in DNA. Predict the likely results in the comic essay for this treatment. Okay, so we know that the reason why we had this tail was because there were these double-stranded breaks that were due to the DNA damage. Well, if the damage that we have is not actually damaging the double strand, if all we're doing is substituting it, what kind of difference would you see? And so you have to think to yourself, if the reason why we have the tail is because of double-stranded breaks and substitutions do not cause a double-stranded break, then that would mean that we're going to see that we're not going to have those breaks. Sorry, trying to rotate. And so it's saying that the undamaged DNA would appear as a round shape called the head. So I should see literally just the head. I would have the head only. You could have said a head with no tail. Another thing that could have been possible was that the tail would be shorter because you wouldn't have as many of the double-stranded breaks from that. Most students usually write this first answer because that's the one that kind of stands out for you. So I hope that the first question was good and that you're making sense. Remember, if you have questions about them, you can put them down in the chat. There's not a lot of y'all in here, so I can see that chat really quickly, and I can easily answer your questions as they come through. Remember, only about the FRQs though. So the second question we're looking at is 2016 number 4. And so with this question, and they give you another diagram, they give you this pretty picture here. And this looks just like a picture that I've actually seen in my textbook. I use the Campbell Biology in Focus, so you probably have a similar textbook. So it says the primary transcript in the figure is 15 kilobases long, and the mature mRNA is 7 kilobases in length. So if I look at my picture, I can see here's my primary transcript, and that one's 15, right? And then I see that my mature mRNA is 7 kilobases. So as a student, I would write on my diagram. I would kind of draw myself up, like on there, say, okay, this is 15, this is 7, because you're probably going to need to know that information when you go back to answer your question. So it says describe the modification that most likely resulted in the 8 kilobase difference. So if we know 15 minus 7 is 8, so they want to know, well, why is this one 15, this one 7? What happened between this picture to this picture? So again, I have no clue. I'm so lost. This makes no sense to me. My teacher didn't teach me this. Like, I don't know any of this. No, I don't know anything, right? So how can we use a diagram to help us? What can I get out of this diagram that would allow me to get the points even though I know nothing, right? So look at your picture. Here when I had 15, I got these black spots and these white spots. Here when it's 7, I only have the black spots. What was cut out? Oh, look, the white spot's gone. And the white spot are called introns. So you get a point for saying that you removed the introns. Seriously? You just got a point for looking at a picture and noticing that there was something that was different. Oh, the white spot's gone and the white spot is intron. So I just said that the introns were removed. See how easy it is to get a point sometimes by just looking at a diagram? Okay. And then it says identify in response to the location in the cell where this change occurs. Okay. So I can see that there's a circle, right? And so if I was looking at a cell, okay, what is a membrane that you have on the inside of the cell in which DNA is found in, right? Well, logically, so going from DNA to some type of transcript, this is all occurring within some type of circle. And I know that DNA is within your nucleus, so I would say that this happened within the nucleus, okay? Now again, that one probably took a little bit of knowledge, but you could have at least gotten that first point knowing that the introns were removed, okay? So the second part says that we need to predict the mature gene mRNA if the full gene is introduced and expressed in prokaryotic cells. Okay, so again, let's give ourselves some little jotting points to say, okay, what do I know about the difference between eukaryotic and prokaryotic cells? Okay, well, prokaryotic cells are smaller, eukaryotic is larger. Doesn't help me there. Okay, prokaryotes do not have a nucleus, and eukaryotes did. Ding, right? We just identified on the part A that this whole processing, this mRNA processing, the cutting out of the introns occurred within the nucleus. So a prokaryote does not have a nucleus. So can it go through mRNA processing? Nope. It has no way to do it, right? And you've probably learned about that transcription and translation actually occur simultaneously. And so what you would say is that it would actually be the exact same length. If we took this gene before it was modified and we put it into a prokaryote, it's actually going to end up being expressed as a full 15 kilobase long part. Why? Well, because of the fact that we can't cut out the introns, okay? mRNA processing does not occur in a prokaryote, okay? So by saying that and identifying and saying, okay, I know the difference between a prokaryote and eukaryote, let me apply that information to this example, okay? You could have also said that it's going to be longer than the mature mRNA because that is a logical thing. They didn't say you had to be exactly 15, but you did have to give a number that was larger than 8, but less than 15. And so, where was I going to go with this? I lost my train of thought. Sorry, y'all. But so I hope that that one was helpful. Again, using the diagram to help you as much as you possibly can. So the last question we have is 2013 number 5. So it gives us this table, and it shows us there's 4 different species, and it shows us these letters, which I know nothing about these letters. Table below shows the amino acid sequence of the carboxyl terminal segment of a conserved polypeptide for 4 different but related species. Each amino acid is represented by a 3-letter abbreviation. Oh, that's what those are. Those are my amino acids. And the amino acid residues in a polypeptide chains are numbered from the amino end to the carboxyl end. Empty cells indicate that there's no amino acid present. Oh, so right here is no amino acid. So you can draw yourself a little note and say, oh, no amino acid. Or you could just say, okay, well, it's blank. Okay, so what do we do from this? So A, thank you, absolute recap. A says, assuming that species 1 is the ancestral species of the group, explain the most likely genetic change that produced the polypeptide in species 2 and the most likely genetic change that produced in species 3. Okay, so I've split this up just because of the fact that it was just too much to have on one slide. So we're just going to look at 2, and then I'll flip the slide and we'll go to species 3. Okay, so if we look just at species 2 and we look along this, we say, okay, these are the same, these are the same, same. Oh, look, those are different. And of course, if you follow it through, you're going to see the rest of them are the same. Okay, so what I see here is that I have had one amino acid that has been substituted for another amino acid. Okay, and I have to explain the genetic change that produced this. Okay, let's think for a second. The change that happened. What happened? One amino acid was substituted for another. So I had an amino acid substitution. I had a mutation. You want to get specific, then that would be a missense mutation, right? Because it was one amino acid that was substituted for another amino acid. And what is this going to do to my whole structure? It could potentially cause a major issue to it, right? But it doesn't ask me to do that, right? It just says to explain the most likely change. Okay, so all I have to do is I just have to say that I had a mutation that occurred at position 4 in which the valine was substituted for the lysine. You could have been all detailed, right? You could have told me it was a missense. You could have told me it was a point mutation. You could have told me it was a substitution. All you really had to say was mutation. Okay. And then the result of it, you could say that there was an amino acid that changed at position 4, and then specifically saying that valine to lysine. Perfectly great. So the second one that we look at was species 3. So if we look between 1 and 3, again, same. Oh, look, we're missing those. So what happens? What causes you to not have an amino acid? Well, that would be another mutation, right? So the mutation that we have here would be a nonsense mutation in which we have an amino acid that has been substituted for a stop codon. And so you wouldn't say that there's a stop codon at 9 and 10. You would say that 9 has been substituted to a stop codon, and then the protein ended, or you would say there's been a premature stop at position 8. Okay. And again, we're looking at a picture. The picture shows us that we no longer have these amino acids here. We had a premature stop. We stopped right here and we don't have any more amino acids. That was all given in the picture, right? And how did we happen? We had a mutation. Okay. You don't even have to tell me what kind it is. Just tell me there's a mutation, a point mutation. Okay. You could have been really super advanced and said that there was maybe a frame shift because we have no idea which one it was. You could say that we had a nonsense mutation. Okay. You could say that there's been something that introduced a stop codon. Okay. That's the big picture here. And how do I know it? Because my protein terminated after valine at position 8. So right here, I no longer see any more amino acids. So there was a termination sequence. Okay, so now I have to predict the effects of a mutation on structure and function of the resulting protein in species 4 and justify your prediction. Okay, now again, remember, they tell you to predict, should never leave it blank. At least try it, right? Who cares if you're wrong? At least put something down. Make a prediction. Guess anything you want to guess. I really don't care what you guess. Just guess something. Sometimes you're right. Sometimes you're wrong. It's okay. So we're looking at 1 and 4, right? And so these ones are the same. Those are the same. Oh no, at 5 we become different. And we're different at all of the rest of the amino acids, okay? So we don't have to know what kind of mutation this is, in which students always want to tell me, right? And you can tell them. There's no problem to tell them there's been a frame shift mutation, that there was some type of insertion or deletion of a 1 or 2 nucleotide pair, and that causes all of the amino acids downstream to be shifted, which causes a different amino acid. Perfectly fine to tell them that. But you don't have to. And none of that is squareable, okay? They just want to know, how does the structure and the function change, okay? Well, because I have different amino acids, what would that do to my structure? Well, if I have polar becoming nonpolar, then it's going to cause the structure to change shape. If I have a change in shape, I have a change of function, okay? That's one prediction. What if maybe we don't have a change in shape? Well, then we don't have a change of function. Then no difference. But maybe that's right. Maybe that's wrong. I don't know. And then you could say that maybe there's a change of shape, but that isn't a large enough change of shape, and maybe we still have the same function. Okay, so there's a lot of different things you probably could have written here, and all of which, guess what? All acceptable. You could have said different shape and different function. Different shape, no change in function. And look, same shape, same function. Okay, so again, all 3 possibilities of things that logically could have happened are all on this paper, right? All of them are scoreable because they can't get in your mind to say, what are you going to predict? So they have to come up with every possible plausible answer that could be a prediction for this because they're asking you to predict. They didn't give you any way to know what was going to happen to that protein. If they gave you some information, you'd have to use that information, but this one didn't tell us anything. All I can see is that my amino acids are different. So I have to make a prediction based on knowing that. And then your justification is just to justify why you said that, right? So there's been a change in the amino acid sequence starting at position 5, and that causes my structure to change, which will then interfere with my function. Changing the amino acid alters the shape. You could talk about binding region, the active site, but maybe the changing of that shape didn't actually modify my active site. Or you could just say that it doesn't alter the protein shape, which thus doesn't change the function, okay? So next week, I will see you again here, same place, possibly same time, who really knows with me. We'll do 2016 number 1 and 2015 number 3. And I hope that was helpful. And I will see y'all later. And remember, your AP Bio Penguins and your Dress for Success. Have a great day. Bye.